All right, last little video clip here about our transcription and translation story. So one thing I want you guys to be able to do is predict the amino acid sequence of a protein if you are given a messenger RNA sequence. Okay, so that means you're being given the messenger RNA. So transcription, the process that made the messenger RNA, has already been completed. So we've already gone from the DNA to the messenger RNA. We just want to do the translation process. And a messenger RNA sequence will look something like this. So you've got a 5' end, that's like the beginning, a 3' end down here, and we've got a series of G and U and C and A, blah, 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 RNA bases in this messenger RNA molecule. So what are you going to do with this type of problem? And these types of problems show up on the TEAS tests from time to time, if not almost all the time. So it's good to be familiar with this. Now, with these types of problems, you are also given a codon table. So it's actually extremely straightforward. You're just reading the codon table and matching a codon on messenger RNA with an amino acid in a protein. So remember, we've talked about how AUG is your start codon. So what's going to happen here is inside the cell, the ribosome is going to attach, but we're not going to initiate translation until we find our AUG, our start codon. So that means all of this is going to be ignored. You're not going to do translation on that. All right, now when the AUG is found, remember you go up here to your table, and AUG specifies an amino acid that's abbreviated as MET, that stands for methionine, or it's FMET in prokaryotes, bacteria. All right, and then you're going to shift over to the next three. CGU, right there. You go to your table, CGU, and incidentally on this table, rows are organized by first letter, UCAG on the table going down. Then you have columns for the second nucleotide base, and finally your third base is organized in rows over there. That's just to help you locate these codons a little bit more quickly. So your CGU here specifies, if you look at your table, CGU, arginine. ARG is the abbreviation for that. All right, and then you would go UGG, look at your table. UGG is right there. That specifies an amino acid called tryptophan, which is abbreviated TRP. Then we would do CAG. CAG specifies right there an amino acid glutamine, which is abbreviated GLN. This is not hard, is it? ACU specifies an amino acid, also specifies an amino acid called threonine, THR, and I'm running out of room, so I'm going to head this way with my protein. And then we have UUU, which specifies phenylalanine. AUU specifies isoleucine right there. ILE is the abbreviation for that. And then we hit UGA. UGA is one of our stop codons right there. Now on this table it is showing you selenocysteine, abbreviated SECYS. That is a special amino acid used in a small number of organisms. So in a small number of organisms, UGA is not used as a stop codon. It specifies an alternative amino acid called selenocysteine. But in most organisms, UGA is going to be a stop codon. And that's it. We stop. So there are no more additional amino acids added to our protein. So this is our polypeptide sequence. That's going to fold up into a three-dimensional shape and become a functional protein, and that's translation. You have gone from this chain of A's and G's and C's and U's on your messenger RNA that has been read on the ribosome. Don't forget that the tRNAs are involved in bringing in the correct amino acids and matching them up with the codons. And you bond those amino acids together, and boom, you've made your protein product. that can go off and do whatever its job is in a cell. So that's pretty straightforward. So when you encounter one of these on a test, just remember you're gonna get something that kind of looks like this. You want to ignore everything until you get to the AUG. Then you start reading your bases 3 at a time off the table. And as soon as you hit a stop codon, it's over. So even if there were some additional bases downstream of the stop codon, you don't read those. Once you hit the stop codon, translation is over. You have made your protein. Do not read these. These are ignored. Those don't get translated. So you want to keep those things in mind. The other thing some students will do is they will start with this and go C, A, G, U, A, G, C. You don't need to do that. The messenger RNA, that doesn't happen. The messenger RNA, this is a real molecule. It's made in a cell, and that's what the ribosome reads, and you pull your amino acids off from that sequence. You don't convert it over to the complementary copy. But I always have a few students who wind up doing that. Don't do that. You just read it directly off of the table. It is find your AUG, read your chain of amino acids, stop when you hit a stop codon, and you're done. You just write out the amino acids and show them as bonded together, and that's your translation product, your protein. That's not hard. Don't mess it up. And I know if you did the Learn Genetics activity from the previous video lecture, they had you go through the transcription process because they were showing you, they wanted you to go through that exercise of, I'm starting with DNA, let's make the RNA copy. And then you moved on and did the translation. I'm not going to require you to do transcription on the exam. You'll just have to do the translation part as a problem, which will count as several points like a discussion question would. Okay. Oh, there will be a laboratory type exercise as well where you'll get some additional practice with these types of problems, and we'll also bring in the concept of mutation with those. All right, so this is a pretty good little figure from your textbook, which is once again just reminding you in a different way of what we've been talking about, what we've learned. So this is supposed to be a little short generic gene on DNA. And they're showing you, you can think about these series of 3 bases being together on the DNA as well. The process of transcription, you make the messenger RNA copy. And again, it's messenger RNA is single-stranded, so you're only really winding up with a copy of one of your 2 DNA strands. And then on a ribosome, tRNAs come in that have these anticodons that can base pair with our codons on our messenger RNA. That's a codon, that's a codon, that's a codon, that's a codon. And the tRNAs are connected to the proper amino acid. So again, these tRNAs are bridging that amino acid to that codon, that one to that codon, that one to that one, that one to that one. All right, if you go back and look at your table, AUG specifies methionine. CUG specifies leucine. So again, it's just showing you how we're going step by step from DNA to RNA to protein and how those tRNAs are involved in between. All right, another thing this means is, from a standpoint of studying genetics and studying proteins, today it's pretty easy for us to get the DNA sequence, the genome, the sequence of the DNA genome for an entire organism pretty quickly. You could have your entire genome sequenced now for about $1,000, so it's going to become more and more common for people to have their genomes sequenced. What do you do with that information? Because actually only a limited portion of that DNA actually codes for proteins. There's a lot of intervening DNA in between genes that don't specify any proteins that are made. But today we can figure out, where on the DNA of an organism, where are the parts that are likely to code for proteins. And if you know this DNA sequence, you can infer from that, you can figure out the messenger RNA sequence, and from there you can figure out your sequence of amino acids that are linked together to build a particular protein. So even if you have never purified or isolated that protein, you've never studied it in any way, you can figure out that it must be there based on the sequence in the DNA, and you can predict the amino acid sequence of that protein, and that would be a first step in starting to understand how that particular protein, what its structure will be, and possibly even what its function could be. Okay, this diagram from your textbook is showing you something that can happen in bacteria. It's basically a transcription translation assembly line. And so what this is showing us is in bacteria, you don't have a nucleus. In eukaryotes, you do. So guess what? If you're a eukaryotic cell like the cells in our body, where's transcription taking place? Transcription is taking place in the nucleus. Translation occurs in the cytoplasm outside the nucleus. So transcription and translation are physically separated by the nuclear envelope or the nuclear membrane in a eukaryotic cell. So in a eukaryote, you got to do transcription in the nucleus. Messenger RNAs go out of the nucleus. They find the ribosomes in the cytoplasm, and they get translated there. In bacteria, though, you don't have a nucleus. So what that means is you can simultaneously do transcription and translation. And that's what they're showing you here. So you got double-stranded DNA in yellow. That's a messenger RNA that's being made, so transcription is taking place. And before that messenger RNA is finished, there's a ribosome that has attached and starts doing protein synthesis. It starts translating that messenger RNA to build the protein. And not only that, but notice over here in yellow, that's a messenger RNA still undergoing transcription. It's still being made off of the DNA. And what they're trying to show you here is that multiple ribosomes can attach, find the start codon, and then scan their way down. Amino acids get pulled in to build the chain of amino acids, or polypeptide. And you can simultaneously have multiple ribosomes moving down a messenger RNA, doing translation and building the polypeptides. Now that's what they're showing you there. That's called a polyribosomal complex, because you have more than one ribosome attached to a messenger RNA at any given time, helping to make multiple copies of that particular protein. Okay. This is actually an image made with an electron microscope that actually zoomed in, and they're able to figure out those little blobs there, which ones are ribosomes, which one is part of the DNA, which ones are RNA polymerase enzymes that are doing transcription. There are techniques to help figure that out when they look at those types of diagrams. All right, just kind of a brief summary, comparison of genetic processes. So we have really talked about three major processes that go on inside cells. And, when you're first studying them, they can be a little bit confusing. One of those was replication. And we talked about DNA replication. Purpose is to duplicate the genome of the cell. And you want to know the starting points and the ending points of these processes. So DNA replication started at an origin on a DNA molecule, and it ends, if the DNA is circular, you actually go back to the origin. You go all the way around the circle, and you finish where you started, or that can also be called the termination point. Transcription starts at a promoter and finishes at a terminator. This is where you make your RNA copy of DNA. That's transcription. And then translation, you go from that messenger RNA copy to the protein, starts at an AUG start codon, ends at a UAA, UAG, or UGA. So you want to know these beginning points and ending points. Try not to confuse the terminology, and know the purposes, what's going on. Why do you have to replicate DNA? Because if a cell is going to divide, each daughter cell has got to have a full set of those DNA genetic blueprints, because if you don't, you can't do transcription and translation to make all your proteins. If you don't have all your genes on the DNA, you're not going to be able to make all those proteins that are essential for your survival. So that's why it's so critical that DNA gets copied before a cell is going to divide. That is your inheritable molecule that's providing all of those genetic blueprints. Okay, so that actually concludes our transcription and translation story. And again, you're going to wind up getting plenty of practice with this on the Learn Smart Labs activities. And I've got my own additional activity I'm going to put in that deals with mutations that will give you some additional practice as well. Just work with the terminology, study it as a step-by-step process, and make sure you know what's going on during each phase, and know your start and your stop places, and you'll do fine with it. The next video clip will probably be pretty short. We're going to talk a little bit about regulation of protein synthesis, which comes down to, okay, just because you have a gene on your DNA, do you really need to make that protein? And whether or not the protein that's being made is determined through genetic regulation. So we'll talk a little bit about that in the 8th video lecture for Chapter 8.